Good evening, everyone, and thank you for joining us for Algae 101. So this is a webinar brought to you by Michigan Sea Grant, along with partners around the state. You'll be hearing from many of them tonight as they speak about their expertise in algae. So first are just a couple of intro slides here. So I wanted to remind everyone that Michigan State University is an affirmative action equal opportunity employer, and our programs are open to all. We also want to acknowledge that the land that Michigan State University and its county offices occupy the ancestral, traditional, and contemporary lands of the Anishinaabe, the Three Fires Confederacy of Ojibwa, Odawa, and Potawatomi peoples. My name is Erica Kleitz, and I'm an extension educator with Michigan Sea Grant based in Southeast Michigan, and I'm glad you're joining us tonight. I did also want to note that we will not be talking about harmful algal blooms tonight. So we are talking about algae, but we're talking about algae that do not produce toxins. So if you are interested in information about harmful algal blooms or HABs, check out our earlier recorded webinar, HABs 101, and also the HABs 101 resources, as we won't be talking about those tonight. We will be talking about algae as a broad category. So the talks that we have tonight include algal ecology, about green algae and cladophora, Didymo, and golden brown algae. So maybe you've heard of these before and maybe you haven't, but either way, you will be learning more about them tonight. So thanks a lot for joining us. And I'm going to introduce our first speaker, who's Julianne Heinlein. So Julianne, you want to go ahead and start sharing?
Hi, everyone. I'm Julianne Heinlein. I'm an aquatic ecologist with the Great Lakes Environmental Center in Traverse City, Michigan, phycologist or an algal ecologist. And my role here is to introduce some very basic algal ecology so that to try to get everybody on the same footing, because it's a variety of backgrounds here and information. So for the people who weren't able to, oh, we're having a little lag time here with my animations. Give me 1 minute here. There we go. So first of all, what are they? Algae is a huge grouping. It's what we call an unnatural grouping. And what that means is that most of the different types of algae are not closely, or large groups of algae are not closely related to each other evolutionarily. In fact, some of the algae is more closely related to us that way than to each other. But they're an ecologically meaningful collection of organisms, loosely grouped by the following list of characteristics. They're all capable of photosynthesis. They have chlorophyll A, which is that green pigment, which leads to photosynthesis. Most of them are aquatic, and unlike plants or true plants, they have no vascular tissue. And what that means is they don't have anything that's specialized to move water up or nutrients around. And so most of them are locked to wet environments. And whole classes, we love to give classes on the different types of algae. And, if anybody's interested in the different types, I would love to help you with that. But today, I'm just going to introduce you to some of the larger groupings based on how the algae live. So who are they in these large groups? First are the phytoplankton. These are the algae that are suspended in the water column, and they have limited mobility. They can move, most of them at a microscopic scale. They have some of them have flagella. They can move through the water. They can regulate air bubbles to move up and down in the water column, but they can't generally move against the current or wave action. So they're at the whim of water movement generally. And they consist primarily of microscopic single-celled group or single-celled taxa, or some of these single cells that live in colonies. And this is, if you're in lakes associations, this is primarily what you're involved with, is phytoplankton. Almost all of the hazardous algal blooms are phytoplankton communities. And these are just some of the many and varied types some lovely pictures here of different things that are living, some common species that are living in your water columns. And a lot of you in lake associations also kind of measure the biomass of phytoplankton by using a Secchi disc. You can see here, going from left to right, we have increased phytoplankton growth, and that kind of obscures, it changes the depth or the clarity of the water. Our next group are the periphyton, and these are those that are attached to surfaces. And I'm going to very briefly go through the different types. First, there's epiphyton. These are algae on plants or other algae. And this is, you're going to see these often in your lakes. You can see them as the brown filmy stuff, or if you touch it, slimy stuff on plants. Often as they grow more and more, they'll color the plants brown, or they'll cover that green color. We have episalmon. These are algae that grow on sand, or on microscopic ones that grow on sand grains. Diatoms often grow on that. We have the epipelon, which grows on soft sediment. And I didn't mention epizootic algae, but there are algae that either have to grow on other organisms or on animals, or can grow on animals. And you can see some of this algae here growing on this dragonfly nymph. And then the last one is, the epilithon. And this is some of the stuff you're going to be familiar with as well. These are all the things that can grow on rocks. Diatoms, blue-greens, green algae, a whole variety of different algae. And then our last group is the macroalgae. These are algae that can grow enough biomass that you can see them with the naked eye. They're large, or they are algae that have plant-like bodies. One of the most familiar that you'll see are the Chara, or the genus Chara, lots of different species, and many people think these are plants, understandably. They're very plant-like. In fact, they're very closely related to land plants. They're not considered true plants because they lack some of the specific characteristics, but they're on the evolutionary line, and they function very much like other aquatic plants. Nidalopsis is another macroalgae that you might see. This is one of those invasive species that we've been dealing with, and it grows a lot like Chara. It can form these huge beds in systems. And then the next two macroalgae that you probably see quite often are cladophora and filamentous green algae. And Marcella is going to take you through a little more specific information concerning those two. So phytoplankton, periphyton, and macroalgae, and of course within those different groups we have a lot more specific and subgroups. So what do algae do in our ecosystems? Well, they photosynthesize, so they are primary producers, and that means they're taking energy from the sun and turning it into sugars and the carbohydrates that basically fuel life on Earth. Just like our plants, our land plants, they do the same thing for aquatic systems and sometimes for terrestrial systems as well, land systems. So they feed our oceans, lakes, and rivers. And some estimates are that they're responsible for about 25 to 50% of all the primary production on Earth. A lot of that taking place in the oceans. And when they go through their photosynthesis, they're using carbon dioxide and water to create these carbohydrates with energy from the sun, and they're releasing oxygen. So they are a supplier of oxygen to the aquatic communities. Fish, insects, other organisms need oxygen dissolved in the water in order to live, just like most of us do. They respire in the same way. So algae, along with the plants in the water, produce some of this oxygen that gets dissolved and is available to other living organisms. It's very important. And the estimate is that marine diatoms, a particular type of algae, are estimated to produce between 20 to 40% of all of the oxygen in Earth's current atmosphere. So it's a big deal. Because they're photosynthesizing and producing these carbohydrates, and it's allowing them to grow and create biomass, they're an important food source in aquatic ecosystems. They feed protozoa and larval fish and other insects, and those in turn feed the lower level fish, and then those fish feed the predatory fish, and then that goes all the way up to birds and other scavengers. So they're very important as primary food sources for these aquatic systems. And you can see this just is some of a variety of the organisms that are feeding on our lovely algae. These guys are filter feeders. This is a Vorticella. It's taking phytoplankton out of the water column, as well as this zebra mussel who filters water. And then we have our aquatic insects and gastropods, even up to this Komodo dragon, which is chomping on some algae. So very important in trophic levels in providing energy for aquatic systems. They're also very important in nutrient cycling in aquatic systems. So they use inorganic chemicals and transform them into organic chemicals, making them available to other organisms and feeding. Nitrogen fixation is one specific type of chemical transformation that some of these do, and actually blue-green algae do the nitrogen fixation. What they do is take atmospheric nitrogen out of the air, which is not available to most plant and algal life. Most of these organisms can't use nitrogen in the form that it's present in the atmosphere. And so
You've got about 5 minutes left.
Oh, okay. So cyanobacteria take those so yeah, I'm gonna move on then. So much to talk about. They're important in carbon storage as well. They stabilize substrates and they provide other habitat for other organisms. So what affects them? Just like any photosynthesizers, light is very important for that. I was going to go through these different things, but I'm running out of time. So light changes based on the type of water, the color of the water, what else is growing in the water, the season. All of those things affect what and how much algae can grow in a water body. Temperature affects how much and what algae are there. We tend to see diatoms in lower temperatures, greens in slightly warmer, and then blue-greens predominate in the warm waters of the summer. So you'll see these successions in your lakes, and a lot of that is temperature based. But nutrients are one of the basic controlling factors of algae, as we know, and I'm not going to get into the others, but nitrogen and phosphorus are two of the most important controllers of algal biomass and what's there. And they're important because of how they're needed, but also because humans have had the most effect on nitrogen and phosphorus through nutrient runoff and atmospheric deposition. And that's affecting what grows, how much of it, and what grows where. And I think I am just gonna stop there instead of getting into a lot of these other details. Too much to talk about.
There certainly is a whole wealth of information out there about algae. Thank you, Julianne. This is Geneva Langland. I'm Michigan Sea Grant's communications editor, and I'm hopping in to help moderate the Q&A. And I see we've got a couple of people dropping questions into the chat. I will nudge you over to the Q&A box in your Zoom toolbar. That makes it easier for us to make sure we're not missing your questions as they come through. So let's start with one. Is warming climate a factor in algal blooms and growth?
Yes, and it can become complicated, and some of the other presenters can jump in as well, but climate change is affecting both the temperature, the maximum temperature of waters, when the seasons, when that water warms up, and also precipitation patterns. So all of those things combined are definitely having an effect on how much blue-green bloom, or how often it happens in water bodies, the amount of biomass, but it also just changes what types of algae can grow and dominate. So we're seeing lots of different effects. It's we can get into more specifics, but short answer, yes, in several ways.
Thank you. A couple folks are wondering about getting copies of the slides. Erica, is that something that we might be able to share out in the follow-up email to participants?
I think you might not have gotten me there. As long as speakers are fine with us sharing a PDF of the slides, we can do that. Otherwise, we will be posting this recording on YouTube, so you can always also pause the recording and take a look at slides more that way as well.
Great, thank you. Let's see, a couple of questions in the Q&A box about more algae growth patterns. Someone is wondering if there's anything that lake associations can do to limit the light coming into their relevant lake to prevent the growth of undesirable algae in the lake.
That's something we can discuss in the hazardous algal blooms discussions, but certainly there are some people who use straw and other floating biological material or riprap to try to control algal growth. It tends to control all photosynthesis, right? Because you're limiting light. So it doesn't just control target species, but there are things that can be done in a non-toxic way. There are natural substances that are used to limit light. And some large reservoirs actually use basically tarps, floating tarps to try to prevent hazardous algal blooms when they're supplying drinking water. But once again, that's something we'll have to get into later, unfortunately.
Thanks. And you mentioned the harmful algal blooms content. Erica just dropped the links to the webinar that we led in March about harmful algal blooms and a resource document with a whole bunch of other links to useful websites or PDFs about harmful algal blooms and the ways that they can affect us in all sorts of different facets. So thank you for that, and we will keep that in mind as a potential topic to bring up in future harmful algal blooms focused webinars. Which algae is the easiest to grow and robust enough in tough conditions like wastewater that might have harmful pollutants?
Oh, that's blue-green algae and green algae are some of the most tolerant species, and they also tend to take up a lot of nutrients when they grow, but it really varies depending on the other factors in the water body. I know that's kind of not an answer, but it becomes complicated, and it does depend on the water chemistry as well.
I can hop in as well. So there's a number of species that are known to be more tolerant. It just kind of depends on what contaminant you're thinking about. Wastewater has a little bit of everything. So I would say there's not a ton of species that are tolerant across all of, everything you might find in wastewater. But an example, Knitsia palea, it's a type of diatom. It's very metal tolerant. It's used in remediation for mines occasionally. So it's very across the board. I think Julie's kind of hinted at this. Algae are very specific in what they thrive in. So across all the different types of algae, yeah.
And I'll pass along a little wink from someone in the chat, said that after mistakenly assuming that some seawall growth was lichen, they stepped on it and found out that it was slippery algae instead. So I guess that's just a note that there are all sorts of interesting things growing on surfaces near water. And if they're slippery, there's a good chance that they're algae. So as you're exploring, please take care.
And probably quite beautiful at the microscopic level too.
Let's just get to a couple more questions before we move to the next speaker. Sue is wondering if you see big blobs of floating algae in a freshwater context, what might those be?
That could be a number of things, but I'm guessing you're probably seeing filamentous green algae, or maybe some large growths of filamentous blue-green algae that probably grew on the bottom, and then as the communities age, they get air bubbles in them and they lift off the bottom and they float around on the surface. And those tend to be kind of like, they look cloud-like, or they're large clumps. That's often what you see as the primary clumps. There are other things as well, though. So if you have a photo of something and you want to send it to me, I'm sure we can share my email address and I can help you try to get an idea of what you're looking at.
Someone in the chat also mentioned colonial bryozoans can look like a floating blob. So are those algae?
Nope, they're bryozoans. Yeah. They're
Yep.
really neat looking, and they can look like a complex algae. And also there are small round colonial filter feeders that grow. They almost look like blue-green blobs with a little greenish color. So there are other things in there that you might be seeing.
Thanks so much, Geneva and Julianne. And our next speaker will be Marcella Domka with the Tip of the Mitt Watershed Council.
I'm Marcella Domka. I am the Water Resources Manager at Tip of the Mitt Watershed Council, located in Petoskey, Michigan. We are an environmental nonprofit focused on protecting our lakes, rivers, streams, and groundwater within our service area. And I've been a connection to Michigan State University as well. I just got my master's in fisheries and wildlife there in 2023. So a bit of a special connection there. And tonight I'll be speaking about green algae, and more specifically, a type of filamentous green algae that Julianne already touched on just a bit, known as cladophora. So without further ado, I will go ahead and get into it. So I'm going to talk a little bit about green algae and algal ecology as well. And I know Julianne mentioned a bit of this already, so I may not touch on some of these points in as much detail, but I'll kind of skim through these pretty quickly. So green algae are, as we know, a group of photosynthetic organisms containing between 9,000 to 12,000 individual species. So there is really a wide variety of algal diversity in this group, which is pretty incredible. They are eukaryotic and autotrophic. So autotrophic means they're using photosynthesis to produce their own energy source. They're using that light energy and functioning as a light harvester to produce carbohydrates. Eukaryotic just means they have a membrane-bound nucleus within their cells. And when I talk about photosynthetic, and Julianne touched on this a bit as well, but they have pigments that allow for this photosynthetic process to happen, known as chlorophyll A and chlorophyll B. And these are actually the pigments that give algae their green color, and hence the name green algae. Green algae range from micro to macroscopic. So microscopic species could include something like Spirogyra, right here on the right, or on the top, I should say, as well as smaller species like Volvox. And then when you think of macroscopic species, some pretty good examples of that would be seaweed or like large kelp forests. Those are examples of green algae, which is pretty neat. And they can be motile or non-motile, which just means they're capable of motion. They come in 4 kind of primary shapes, I should say, 4 kind of categories there. They can be unicellular, just consisting of one cell, forming these large colonies, hence the term colonial, tubular or filamentous. And I'm going to focus primarily on filamentous algae, since cladophora is filamentous, meaning it has these long, fine hairs and has that hairy texture to it. And while they are typically found in freshwater ecosystems, there are terrestrial and marine species of green algae. So once again, Julianne focused on this just a bit, so I'll go through this quickly, but we know that green algae are really critical components of freshwater ecosystems. they form the base of food chains as primary producers, and just about every other organism is relying on algae as the base of these aquatic food webs. They uptake carbon dioxide from our environment, and they generate oxygen as a byproduct. And the generation of this oxygen contributes to the success of fish and other wildlife, and really ensures that the ecosystem in question is stable. They also provide shelter and food to organisms like aquatic insects, macroinvertebrates, and fish. And humans use them as well. So we use algae for a food source, and we use them in our pharmaceutical industry as well. So the really interesting part about green algae is that it's inherently linked to water quality. And a lot of what I do as the water resources manager here at the Watershed Council is looking into the connections of algae, nutrient pollution, human activities, and how those can influence water quality. So green algae are highly susceptible to environmental changes, meaning they can function as bioindicators of water quality. They can tell us whether that water quality is great or maybe not so great. So what I mean when I say a bioindicator is that when we have excess growth of green algae, particularly cladophora, which I'll get into in a few slides, that can indicate that we have elevated nutrient concentrations in our water bodies. So by nutrients, I'm referring to total phosphorus and total nitrogen, or different species of phosphorus and nitrogen. And so while nitrogen and phosphorus are essential elements that are needed in all aquatic ecosystems, they're often present in excess from human-induced or anthropogenic activities and can pose a threat to water quality. And when we have algae that is overgrowing in response to human activities, we can see physical, chemical, and biological consequences from that overgrowth. So to elaborate on that, algae can, produce some problems if it's present in excess. And some of those problems are physical, meaning the algae could clog water intake pipes or filters. It can impact recreational activities like fishing, swimming, and boating. And on top of all that, it's just unsightly. A lot of people, don't like to see algae on the lakefront or don't like to swim with algae. It can have kind of a, a slimy feeling, or it may, impact the aesthetics of a property. Chemical impacts of algae overgrowth can include hypoxia. So when algae break down in an ecosystem, they require significant oxygen. And I think that's something Julianne touched on, that many species require quite a bit of oxygen. So when they're using all of that dissolved oxygen in water, they're kind of creating an oxygen imbalance, which can lead to dead zones or zones of very low oxygen, known as hypoxic zones. And finally, when we're referring to biological effects, we can see when water becomes really eutrophic, meaning it has high nutrient levels, high algae, probably low water clarity, we will see high algal biomass, and that can cause quite a bit of issues, ranging from decreased water clarity. So that is represented on the bottom left by a Secchi disk, which is what we use to measure water clarity or transparency. Depletion of dissolved oxygen, which can be an issue for wildlife, as the less DO you have, the less you have available for species that live in the water. Mucky lake bottoms, and that kind of connects to water clarity as well. A lot of residents, especially in northern Michigan, really value clean, clear lakes. Clear water doesn't always indicate clean water, but aesthetically speaking, that's a very valuable resource, is those clear lakes and those clear lake bottoms. Eutrophication or overgrowth of algae can cause instability of food chains, as well as death of benthic organisms. And that can often occur when there's that depletion of dissolved oxygen, and organisms that are on the bottom, or like the benthic zone of a lake, may not be getting enough oxygen to survive. What I really want to emphasize here is that green algae and cladophora are not inherently problematic. They're naturally present in freshwater ecosystems, but these consequences shown on this slide here is they're not due to the existence of algae, but rather results from green algae's response to human-induced impacts. So after kind of going over all of these potential water quality issues caused by overgrowth of algae or eutrophication, one type of this algae that can be indicative of water quality issues specifically is cladophora. So cladophora, what is it exactly? So it's clearly a type of green algae. It's filamentous, meaning it is long with a very hairy texture. And let me show had a photo on an earlier slide here. This is not the best photo down here, but on the bottom there, it's just a very hairy green texture there. Sorry about that. Let me move forward here. So it typically attaches to substrate such as wood or rock within a lake, and it is known to bloom in response to excess nutrients. So it's difficult to tell based on maybe the presence of cladophora alone what the type of freshwater quality issue is that you're having, because it can indicate a variety of things. It may be septic system malfunction or failure, where you're having nutrients, leach directly from a lakefront property into a lake, agricultural runoff, or it could even bloom in response to zebra mussels, quagga mussels, or low lake levels. So there's kind of a variety of factors there. So why should we care about the kind of excess cladophora blooms and how is it relevant to my work specifically? Well, a lot of the work we do here, we conduct shoreline surveys, water clarity and quality monitoring. We look at different lakes and lakefront properties to understand if there may be nutrient issues from any of these issues that I mentioned on the left here, maybe some septic system issues, agricultural runoff from a tributary nearby. Maybe we have invasive species like zebra or quagga mussels, or differences in lake levels. And shoreline surveys, water clarity and quality monitoring, that all kind of aims to understand why algae is behaving the way that it is and how it's responding to human-induced impacts.
So you've got about 5 minutes left.
Perfect. And by collecting this long-term water quality data and by monitoring algal concentrations across different lakes in our service area, we can understand and maybe predict long-term environmental changes. And that's why, algae is such a relevant part of a lot of environmental work that we do up here. So ending on a more positive note, obviously algae is a very critical part of our ecosystem, but it can produce some impacts when it's blooming in excess. So there are different things that you can do. And I like to emphasize that you should apply best management practices or BMPs to your personal life. So this may not be relevant to your day-to-day life, but maybe some of these things are especially relevant to you if you're a lakefront owner or something like that. So mindfully applying fertilizer, not applying fertilizer with phosphorus in excess, practicing regular septic system maintenance. So we recommend every 3 to 5 years for an inspection to make sure that your septic system is properly functioning. Sometimes they're in need of replacement parts. Greenbelt installation, that's something that is one of my favorites to talk about. A greenbelt is essentially a strip of native vegetation. It's pictured up here at the top. And those are often planted on lakefront properties or on areas of elevation to collect any runoff, allow this runoff to infiltrate the soil, collect excess nutrients, stabilize shore banks to prevent erosion. And something that we do here at the Watershed Council is we offer greenbelt installations for folks wanting to kind of reshore their shorelines and prevent excess algal growth. Something else, rain gardens, native species plantings, that can all prevent this excess algae growth. It can prevent scum and these big cladophora mats from forming. And one last thing is to not dispose of any organic matter in storm drains that can contribute to further algal growth and algal blooms. So there are a lot of things that you as an individual or a homeowner can kind of adopt into your personal life to prevent any excess growth. And we want to continue to keep algae a critical but balanced part of our freshwater ecosystem so that it's not present in excess and causing all these water quality issues. references there. And that's all I have for you tonight. It looks like it ended a little bit early, but I'm happy to answer any questions.
Thanks so much, Marcella. This is
Yeah.
Geneva from Michigan Sea Grant again with some more questions from the box. And keep them rolling if you've got specific questions for Marcella or for our group of panelists. We have someone wondering, just to clarify, algal blooms can happen in lakes without human inputs, or, well, let me make that into a question.
Yeah.
Can algal blooms happen in lakes without human inputs?
Yeah, because cyanotoxins are naturally occurring. A harmful algal bloom can be exacerbated by human impacts, but it can occur naturally.
And not all algae species that respond to excess nutrients are necessarily harmful ones, right?
Correct. Cladophora does not contain cyanotoxins. It's not harmful to swim in or touch. We look at Cladophora purely as like an indicator of septic system malfunction or excess nutrients, but it's not, Cladophora itself is not a harmful algal bloom. Great question.
Thank you. Let's see, we got a few more in here. So we know that over the course of a year, the types of algae that grow in an inland lake are going to fluctuate and change.
Mm-hmm.
If someone is trying to understand sort of the algae profile that their lake is experiencing, how often might they see things turn over? And this is specifically from someone wondering how often water quality might need to be assessed throughout the span of a recreation season.
Yeah, I have like a guess. I don't know if it's an entirely accurate answer, but I would kind of line up water quality and algae monitoring with lake stratification. So we have a stratification period for freshwater ecosystems over the summer, where you have these temperature layers. And you might see, based on differences in temperature and available sunlight, you may see different species of algae. You're probably going to see a lot more growth in the warmer months when algae can respond to that sunlight, respond to those warmer temperatures. So I would almost line up that monitoring with lake stratification and seasons. I don't know if any speakers want to chime in there, but those would be my initial thoughts there. If no one's chiming in, maybe I answered it correctly.
Often we do when we're doing biomonitoring, we'll do a spring, summer, and fall
Okay. Yeah.
collection. And it is often related to temperature changes as well as changes in the nutrient levels in the different strata of the lake, the different layers. So it's, yeah, seasonal, but related to a lot of things that change with season.
Yeah, absolutely.
Got a question about how to choose the plants that can be a good fit for a rain garden or a green belt. The question asker specifically mentions bulrush, but I guess to expand that generally, are there places people can go to get that kind of information?
Definitely, yeah. My email is listed here in my direct office line. If you just send me an email, we have some great resources at the Watershed Council that I can send you. We emphasize that they should be native plants that have long root systems that can prevent erosion and keep those shorelines completely stabilized. And those long root systems are more effective at in uptaking nutrients and preventing that from entering our lakes. So I am not the plant expert on staff, but I definitely have some resources for some native species that we recommend for both greenbelts and rain gardens. And often there's quite a bit of overlap as well.
And I'll just add, in addition to contacting Marcella, you can look for your local county conservation district or a watershed management group, or your local county extension office can often point you in directions for where to get native plants locally and how to get local advice for what plants might be a good fit for your property area.
All right, next I'm going to turn it over to Jordan Stoll. Jordan, I'm going to let you go ahead and share your screen, and Jordan is with Trout Unlimited.
Perfect. Well, thank you so much for having me and for being here. It's always exciting to tell people about algae because most people aren't very excited about algae usually. So thank you for listening to us. My name's Jordan Stahl. I'm an aquatic ecologist with Michigan Trout Unlimited. Today I'm going to talk to you a little bit about Didymo, some of the work that we're doing across the state and how you can get involved. Oh, and I'm not progressing. Oh, there we go. Okay. So Didymo or Didymosphenia geminata is a variety of diatom, which is a type of algae. Diatoms are pretty cool in that they build their cell walls out of glass. They're very ornate and beautiful when you look at them under a microscope. If you've ever walked around a stream and noticed that there's kind of a biofilm or a glimmer on the rocks or the surface of the stream that's kind of golden brown, it's probably a biofilm that's largely dominated by diatoms. It's often usually very slippery too, as someone else found out, it sounds like in the chat. They often thrive in cold water, high light environments. So oftentimes in winter, early spring, before the leaves come on, there's it's kind of prime time for diatoms to thrive. They're favorable fish and invertebrate food. So diatoms are unique in that when they photosynthesize, they store their photosynthate or their energy in lipids rather than starches. So you can think of diatoms more like avocados, whereas a lot of other algae, like your green algae, are more like potatoes. Avocados, being very nutritious and lipid rich. And diatoms are unique in that, because they make those glass cell walls, they don't decompose as quickly as other algae. They actually can be stored in the sediment in the bottom of lakes. And because of that, you can take sediment cores of lakes and use them to look at the fossils of algae throughout history, so to speak. And because certain diatoms grow under certain conditions, you can start to get at what conditions might have been like in a lake years ago compared to now, just based off of changes in the diatoms that are present. So overall, diatoms are super neat, in my opinion. Hopefully you think that as well after this talk. But like you've heard from other speakers, there can be some consequences when they don't behave. So Didymosphenia geminata, or Didymo, or rock snot, all the same thing, is a variety of diatom again that causes problems when it blooms. So all this information on the slide is directly from the invasive species info.gov website. In the state of Michigan, Didymo is considered an invasive species. People debate whether it's invasive or not. For my personal opinion, I don't really care. It's kind of semantics, but it is a nuisance species. And if we keep calling it invasive, we can keep getting money to better understand it. So I'm happy to keep calling it invasive for now. It's been around for a while, but it really hasn't started causing problems until recently. So I'll jump into a little bit more on the issues it can cause here soon. But just its appearance. So this picture is taken on the upper Manistee back in, I think, 2022. Oftentimes, Didymo blooms people describe them as kind of looking like toilet paper floating around the bottom of a stream. They're wispy. They're not that bright green algae that, like cladophora, like Marcella was showing you. They're kind of more golden brown to gray in appearance. You can tell the difference from Didymo blooms and other algae by its texture as well. So if you were to walk through a river and spot something like this and to feel it, people usually describe it as feeling more like wet wool rather than being slimy. So it's just a little bit different from your typical algae bloom in more ways than one, but that's one. So in the state of Michigan, Didymo is confirmed on the Boardman River, the Manistee, the Ausable, and the St. Mary's. It hasn't bloomed on the Ausable. However, since we've started sort of looking for it, we're finding that it exists in the background community. And I'll talk a little bit more about what it means to bloom. But in these pictures, this is examples of Didymo blooming, and that it's growing in a large biomass covering the bottom of a stream. Okay. So this picture is from a Didymo bloom in New Zealand, where Didymo is most definitely invasive. You can see that just how much biomass it can accrue at the bottom of a river. you imagine, the bottom of a river typically being sandy or, gravelly and having, really great habitat. When Didymo grows and turns it into, a giant bed of slime, very homogeneous, you can imagine the effect it would have on habitat for all the organisms in that river. But just to kind of emphasize, and Marcella did this too, and that poll at the beginning also kind of touches on this. We lately have been talking about algae in a negative way, and that we kind of think about it when it causes a problem. But overall, algae are super critical. Diatoms are the base of aquatic food webs. They're essential to our stream ecosystem. So we shouldn't overall dislike diatoms or algae. We should recognize that they can be a problem occasionally, but overall are really critical. So just to get back to Didymo, some of its unique ecology. So when you think about harmful algal blooms and a lot of other algae, they generally bloom when there's a lot of nutrients. So if you think of them like plants, you fertilize your plant, they grow like crazy. Didymo's the exact opposite. It seems to consistently bloom or form, these stalks and grow in this crazy high biomass under really low bioavailable phosphorus, which is just phosphorus that's floating around in the water and able to be taken up by the algae cells. So when that's really sparse, that's when these little buggers go crazy. They also seem to bloom under high light conditions. So when there's plenty of light, the cells are capable of photosynthesizing and growing like crazy. They also seem to like consistent flow. So Didymo blooms are often found downstream of dams that have are kind of consistently releasing cold water from the bottom, and not they like cold water. So like I mentioned earlier, diatoms in general are kind of tend to favor cold water, and Didymo is no exception to that. I say all of this, but not always, because there are some instances where Didymo's been found not under these conditions. So this is a figure from a paper from a study that was done in Connecticut. So just to orient you to this figure, on the bottom, you have concentration of phosphorus being low, medium, and high. And then you have the frequency of Didymo being found in specifically Didymo forming stalks. So the frequency is the number of streams sampled. So at really low phosphorus conditions, they found that in 4 streams, they found Didymo forming stalks. 2 other streams, Didymo was present, but it wasn't forming those stalks. At kind of, still low, but between 5 and 10 micrograms per liter of phosphorus, Didymo's present just in the background biofilm community, but it's not causing problems. It's not forming those stalks or blooming. And then above about 10 micrograms per liter, Didymo doesn't seem to be, present. It's being outcompeted by other algae. It's just not there. And this is a picture of the upper Manistee, where Didymo has been found to bloom. Again, it usually blooms under 5 micrograms per liter of phosphorus in Connecticut, where this other study was done. But on the Manistee, there's some evidence that it might be blooming at higher concentrations than that. So Lake Superior State University has taken some samples to kind of look at water quality and phosphorus alongside when Didymo's blooming. And there's evidence that Didymo's bloomed around 10 micrograms per liter in the upper Manistee. But more data and more investigations needed to really understand what levels of phosphorus might control or support Didymo blooming. In general, Didymo has a lot of effects on the stream ecosystems. I won't go into too much depth on this, but like I kind of mentioned earlier, it reduces that habitat diversity. So instead of having different substrates and you can have a system that's just the entire stream bed's covered in Didymo. It clogs up the benthic habitat, and then there's also consequences to mat decomposition, just like how Marcella was explaining to you earlier, how when algae decompose, that process sucks out the oxygen from the water and can cause kind of dead zones or low oxygen zones. Didymo can also negatively impact the macroinvertebrates that are present. So these 3 taxa here are your EPT, or your bioindicators for good healthy streams. So your Ephemeroptera, your mayflies, your Plecoptera or your stoneflies, and your Trichoptera, your caddisflies. all three of these groups, generally you see lower diversity and abundance of them when you have Didymo blooms. You'll see other macroinvertebrates like chironomids and gastropods that do better in Didymo when Didymo's around, but these more sensitive taxa tend to become less abundant, which is unfortunate because they are more of the favored food for a lot of the fish that people tend to be drawn to, such as trout.
And you've got about
Okay,
5 minutes left, Jordan.
Perfect. Thank you so much. So what is Trout Unlimited doing? So generally speaking, our mission is to be proactive across, all cold water conservation topics. Right now, we're trying to monitor the spread across the state. So again, we know it's bloomed in 3 rivers. It's at least present in the Au Sable, but not blooming. So we're trying to better understand the distribution across the state. We're also trying to better understand sort of the habitat conditions and water quality that might be supporting, or not supporting Didymo. We did receive grant funding to expand our initial effort on this. So this effort thus far has sort of been ad hoc, but is going to be more formalized starting in October. So if you were interested and want more information on this, please reach out to me. I'll be sending people sample kits, hosting all sorts of education outreach events, and creating a network of folks who are interested in Didymo to try to sample across the state, get folks to send me samples so I can determine if Didymo is present or not. Yeah, so please contact me if you're interested in participating in this at all. And here's a QR code to use Didymo monitoring website for more information. It also has this Didymo monitoring form that you see on the slide. There's also, I believe, a link in the chat too. So the 3 questions that we're trying to get at with this grant, and sorry, I keep having to move my little pictures of the host around here, but our key questions are, so at what concentrations of phosphorus does Didymo stock formation begin in Michigan rivers? So kind of what I was getting at earlier, where it seems like somewhere between 5 and 10 is where Didymo's present, and less than that is where, Didymo stock formation seems to happen elsewhere. So we want to know if that's the same in Michigan. Are there other environmental drivers that are important? So things like, different substrate type, temperature, conductivity, flow. So we'll be collecting all of this information on the Manistee, Boardman, and Au Sable to try to get at that. And then, just trying to understand if Didymo is present in more places across the state and just not blooming. So, unless you take a sample and look at it under a microscope, you don't really know if it's there. So again, we're just trying to collect samples everywhere across all times of year to better understand where it's at across the state. So here's a map of all of our samples so far. The green dots are samples that did not contain Didymo. The red dots are samples that did contain Didymo. And so just, what you can do, you can definitely tell people about Didymo, make them aware. You can send me samples and become involved. You can encourage folks to decontaminate their gear. So a couple ways to do that. You can freeze your gear. So if you have a chest freezer, you can throw your waders and any fishing gear that would fit into a trash bag, throw it in your chest freezer overnight. 409 solution is known to work for Didymo. There's a paper that will be coming out soon that demonstrates pretty conclusively that Didymo works to that 409 solution works to decontaminate Didymo from gear. But I think most importantly, it's just spreading the word, making people aware of this. There's a lot of people, I think, that go fishing or that come in from other places in the world that aren't aware of invasive species, and that invasive species can be spread on angling gear. So, education, outreach, spreading the word, really critical. And without further ado, if anyone has any questions, please shoot me an email. More than happy to, engage with any groups who are interested in having me come to talk about Didymo in a little bit more depth, and to, distribute some sample kits if folks are interested in sending me samples and, knowing if Didymo's in your streams or not. So thank you.
Thank you so much, Jordan. I've got several questions for you popping up in the Q&A box. Someone is wondering what the sampling landscape looks like in the UP.
So I've only taken 4 samples up there. I didn't find Didymo in any of them. Folks want to take samples, they can. So the process is pretty much going out to a stream with a toothbrush and some sample containers and scrubbing rocks or wood, whatever, substrates available, dabbing it in some water, putting in a container with some preservative and sending it off to me. Has that answered their question?
Yeah, we'll just make this an invitation for folks who are in the Upper Peninsula.
Yeah.
Get in touch with Jordan and get a collection kit. Add to the
Yep.
data.
Yep. And I'll be up there in a few weeks, so get a hold of me sooner than later, and I can maybe give you some supplies sooner than later.
Ah, there we go. So we know that Didymo is particularly a problem in rivers. Does Didymo show up in inland lakes?
As a blooming algae, not that I'm aware of. I don't know if anyone else has seen it as a blooming algae there. I know that cells have been found in lakes, that just as washed through being deposited in the lake sediments. But I'm not familiar with it being a blooming issue. Didymo is generally like Julie mentioned, an epilithic, epiphytic. It grows on surfaces. So it's not usually just a free floating diatom. It's pretty bulky and heavy. It usually sinks out of water.
And that's a good reminder that just because it's present doesn't mean that it's necessarily a problem or going to be a problem. But with something like Didymo, where we're still trying to figure out where it is and what it can do, we definitely want to know if it's present.
Definitely. Yep. Yep. Yeah, it seems like the conditions have to be right, and the conditions, as far as we can tell right now, are low phosphorus, high light, cold water, clear water. So but there might be other things in there too. I've just recently saw a paper about conductivity, potentially, water that's exposed to salt water or salt from, like road stream crossings. Most likely wouldn't have a Didymo bloom problem because Didymo maybe doesn't thrive under higher conductivity levels. So
Mm-hmm. Always new things to learn about these things that we share our lakes and streams with.
yes.
I'm going to ask this as a selfish plug way to tee Erica up to talk about one of our programs. Can Didymo be spread from one water body to another from boats that haven't been cleaned? And what can we do about that?
Yes, absolutely can. So we think that Didymo is being spread by angling and recreational use. It can live on gear, especially damp gear like felt sole waders, for up to 40 days out of water. If you are a person with a boat, you can take it to a car wash and spray it off in a car wash. That's a great way to decontaminate. Hit it with some 409, spray it off. You should be good to go. Yeah, definitely decontaminate your gear.
Great. Did you have anything else you want to share at this point, Geneva? Or
Well, thank you, Erica, for dropping the link into the chat or the information into the chat for the Intro to Lakes course. That's a really awesome MSU, Michigan State Extension resource about inland lake management. And I wanted to tee you up to talk about My Paddle Steward as another resource for people to learn about how to clean their boats and equipment.
thanks. Yeah. So we do have a program called My Paddle Stewards where we teach people using non-motorized boats, so kayaks, paddleboards, and similar, how to decontaminate their gear as well, because those smaller boats can still spread things like Didymo and other things that are problematic. It's not even just plants and things, it can be fish diseases. So there's a lot of things, unfortunately, that we humans move around faster than they can move on their own. So anytime you can decontaminate, or the longer you can leave stuff out of the water before you enter a new water body is always a good plan. And I'll make sure there's a link about My Paddle Stewards that gets in the chat. I did want to reiterate, especially for people that are asking questions about nutrients in lakes. Lakes are they're complicated in terms of like, what are the sources of nutrients? What is the nutrient cycling that's happening? What are the aquatic plants? What's the algae present? So great questions coming in about that. If you want to take a deeper dive, the Intro to Lakes course is a great way to do that. But there's also been some other links put in chat, and we also have resources within like MSU Extension. There's also the MyCore monitoring program where you can learn more about what's in your lake and what the current conditions are. So check out some of those resources if you want to learn a little bit more about your lake. And then maybe you can take a look, Jordan, when you get off. There's a question, well, a comment in the chat about a potential new location for Didymo. So I'll let you take a look at that. And otherwise,
yeah, if anyone sees any suspicious algae that you think is Didymo, give me a call, shoot me an email. Happy to come and check it out, or at least receive some samples from you. The other thing I wanted to note is that 409 also works for New Zealand mud snails. So you get, the benefit of taking care of both of those invasive species with one product, which is convenient. So
yeah, especially since those are both found in the same general areas, at least both kind of northern lower Michigan. So yeah, awesome.
Thanks
everyone. So
much,
much, Jordan. And our last speaker of the evening will be Jan Stevenson.
Yeah, good evening, everybody. So my name's Jan Stevenson. I've worked at Michigan State University and the University of Louisville for many years, and I've been an algal ecologist for about 50. And the last number of years, I've been working on a rather novel problem in northern Michigan on golden brown algae. And now that I'm retired, that's one of the projects that I'm going to continue to kind of work on, as well as a few others to keep myself entertained. So the golden brown algae, there are whole groups of algae referred to as the golden brown algae, like there are the green algae. The diatoms are actually one of the groups of the golden brown algae. And but I'm using the phrase this evening with this presentation to refer to the nuisance growths on the bottoms of lakes. Typically in this situation, really low nutrient lakes. And these kinds of growths are really common in all kinds of water bodies, but it's really uncommon for them to occur in these low nutrient lakes. So the people in the Torch Lake area, the watershed association that I'm working with, they've referred to this as golden brown algae on the bottom of our lakes. This is a problem, and we refer to it as GBA. So you'll hear that phrase. So this slide illustrates the problem. Back in 2010, aerial photographs show that there was really no algae growing on the sands. We have these beautiful white sands that are surrounding the lake in these shallow areas before you hit the drop-off, which is the deeper blue. And then around 2015 and since, these shallow water areas have been turned golden brown by this thick mat of algae that's growing on the bottom. And these are very low nutrient lakes where this is happening, with phosphorus down in the 2 to 4 micrograms per liter range, and nitrogen in the 3 to 400 microgram per liter range. So they have very high water quality. And we've been working on the problem for about a little over 10 years. And it's different than a lot of other situations where we're not talking about algae in the water column. We're talking about algae down on the bottom. And I'm not going to go through the kind of timeline of what's gone on, but it's been, it's involved working very closely with a watershed association. And then more recently, I've kind of taken over the study design to kind of expedite the work and focus on some key questions. So I'm going to describe what GBA is, where we started with the research, some of those key findings, a new hypothesis associated with low phosphorus, and then what we're doing now. Before I do that, I'd really like to thank the Three Lakes Association for funding a lot of the work, particularly the water chemistry analyses. And there are specific individuals like Dean Branson and Becky Norris, Trish Narwhal, and a group that have spent a lot of time going out in the field and doing the sampling. There are also a bunch of other people associated with the University of Michigan and Michigan State University that have helped along the way. So first I want to describe the macro structure of these mats. They're about a quarter-inch to a half-inch thick. They're sitting on the bottom of the lake. The surface of the mat has, in the upper left-hand corner, the surface of the mat has a thick, dense algal mat, and then that's underlain by a lot of calcium carbonate. And as we start to focus in on it and higher magnifications versus just on a plate in water, we see that the very top of the crust is composed of cyanobacteria. And then over on the right here, these are a lower magnification figure of rafts of diatoms that are loosely associated with this. And one of the key diatoms over here on the right is Encyonema everglanianum. It's found really commonly in low nutrient conditions in the Everglades. So down on the left, we see other diatoms also that are floating around the mat. And as I tease apart this mat and get away from the crust, which again is covered with cyanobacteria, and the golden brown pigments are found actually in the mucilages over the cyanobacteria and the chloroplasts of the diatoms. And that's where the golden brown color comes from. The calcium carbonate that's under that is very white and granular, but there are a lot of other cyanobacteria, filamentocyanobacteria, as well as diatoms buried in that, and really healthy, buried down in this calcium carbonate matrix underneath it. So in starting out back in 2013, Rex Lau, Pat Kaselek, and I, as well as a group of other Lake Association people, brainstormed hypotheses that could potentially cause this. So we have a long list of hypotheses that we're testing, and we continue to consider those as we narrow them down. But we had to, because of budgets, we had to narrow down what we were going to focus on, basically on plausibility of the cause. And one of the ones that we thought was most likely was groundwater contamination. So that's where we started the research, and I'll explain a little bit of that to you in this next example of how we went about that. So we did surveys around the lake, so 20 sites around Torch Lake, and took samples of benthic algae using a sled that's basically about the only way to take samples. And at the same time, we took GoPro videos to look at how much golden brown mat is on the bottom and get some idea of the structure as it was being scooped up into the scoop. We also used aerial photographs to take pictures around the lake and watch the seasonal development of the mats from the beginning, basically from May through September. And what we were expecting to see was a relationship between where the most houses were around the lake and where we were getting mats. And basically, we were finding mats pretty much around the entire lake. So this did not support groundwater being a source of nutrients causing this, because we'd expect there to be a relationship between sources of nutrients, for example, houses or agriculture, and where we saw the GPA occurring. So we went back to one of the original questions that we had is, so what's changed in the lake that could possibly be causing this kind of a problem? And so I during one of the reports, during the development of a 2021 report, I went and got data from the CLMP and the Tip of the Mitt watershed databases. And what I found was that there was a decrease in total phosphorus in Torch Lake over the last 20 or 30 years. In fact, there's evidence that that's actually occurred over the last, doing the calculation in my head, 75 years. Not just the short term over which we have these, the actual measurements from these watershed groups. So there's debate about whether these trends are reliable and whether the data are reliable. But there's pretty strong evidence for it, because if we look at lakes across northern Michigan, we find out that most of the big lakes have significant relationships with decreases in phosphorus over the last 20 or 30 years in the water columns of the lakes. So then the question becomes, how could you have a decrease in phosphorus cause more algae in a situation in the bottom of the lake?
Hi, Jan, you've got about 5 minutes left.
Okay, that works. Thanks. So we're thinking maybe this low phosphorus is causing it. Well, there are two really good examples of this. One a little bit better than the other. One of them is the one that Jordan just talked about, is Didymo. The Didymo blooms occur in low phosphorus conditions in streams. They don't occur in high phosphorus conditions. So phosphorus is somehow having a negative effect on Didymo. And there are a number of hypotheses about how that's possible. The other really in Everglades, where I've worked for, again, doing the calculation, 30 years, there's a calcareous algal mat that develops in the Everglades in the natural background condition of the Everglades. It's a thick calcareous algal mat, and when phosphorus pollution occurs, that calcareous algal mat disappears. And we've done, many people have been studying this for many years to set up phosphorus management targets. But one of the key species in this is this Ensignium evergladianum that we see in the benthic algal mats in Torch Lake. So that has steered us toward this low phosphorus hypothesis, that it's possible that low phosphorus is causing that. I mean, it causes calcareous algal mats in the Everglades. So could it be causing that in these northern Michigan lakes? So we've been continuing to do these 20-site surveys in Torch Lake, basically across the season. And another kind of survey approach that we're using is actually surveying a number of the lakes in northern Michigan. And we're finding these benthic algal mats tend to occur in the low phosphorus lakes, Charlevoix, Elk, Lake Bel Air, some of the lowest phosphorus lakes. So we're finding a statistical relationship with low phosphorus, but could there be something else associated with low phosphorus that's actually causing the growth of these mats? So to evaluate that, we're using experiments where we manipulate phosphorus condition using these frames, these tubular frames that leak nutrients and enrich the central square area and some of the surrounding area. So we can increase phosphorus, increase nitrogen, and kind of manipulate conditions that way. And we've done the same kind of thing in a bowl, which gives us a slightly different perspective on the changes in environmental conditions that we can manipulate. So we've been using a couple of different experimental approaches to show that, to test that hypothesis. Does low phosphorus, if we increase phosphorus, do we get a change in the algal mats that would actually look more like the calcareous algal mats, the GBA mats, or is it a decrease in phosphorus? Is it an increase or decrease in phosphorus, or is it an increase and decrease in nitrogen that's causing those problems? So again, at this point, it really looks from experimental and the survey data like low phosphorus conditions are causing these golden brown algal mats to occur in the lakes. But we're far short of actually having enough causal evidence to move to management strategies that would change the conditions. In fact, it's kind of difficult to come up with management strategies that you would think would change those conditions. But I do have some, speculative thoughts on what's going to happen in the future associated with this. But our next steps are to continue this work and really finish analyzing samples and analyzing data, and actually run a couple more experiments to tease out these relationships to determine what the cause, actually at this stage, determine whether low phosphorus conditions could be causing these mats, or, and if that's not the case, then what else could it be? Thanks. If you'd like to look at, learn a little bit more about this, there are a couple links that I've provided for reports and a YouTube video that actually describes more extensively these results.
Excellent, thank you. And we can make sure that these links get out in the follow-up email to folks who registered for the webinar. Folks have been dropping a couple links and pieces of information into the chat, and Jordan had an interesting little side note about why Didymo and GBA may both do well in low phosphorus environments. And essentially the idea is that the stalk formation in Didymo allows them to, sorry, my chat window keeps scrolling around in funny ways and it's hard to read. There we go. The stalk formation in Didymo might allow the diatom to move higher into the water column, exposing itself to more dissolved phosphorus in the water column than when it lays flat on the bottom of the stream. This stalk may also allow Didymo to process organic forms of phosphorus found in water. When phosphorus levels increase, other algae may thrive and outcompete Didymo and GBA. So essentially the idea, it sounds like, is these two types of algae might just be really good at exploiting that low phosphorus niche. And then when there's more phosphorus around, other species get to have their heyday and kind of elbow out the GBA and Didymo. Does that sound like a good summary of what you put in there, Jordan?
Yeah, yep. Yeah, there's also probably some other variables that we don't fully understand yet too. More research is needed. My guess is that, there could be something with micronutrients, there could be something with conductivity, there could be something with pH, there's a number of things. It seems like it just, you have to have the right sort of variables aligning for these blooms to happen. And they're pretty much the polar opposite of, something like cladophora that's going to grow when there's plenty of phosphorus around. So
thank you.
the stalks are made out of a polysaccharide.
Oh, good. Yes, thank you. That was another question from the chat. It looks like Jan might have dropped out of the Zoom. So maybe he'll be able to join in another moment. But we do have a few questions that could be for the group at large. Do we know why, well, someone wondering why Glen Lake, which is a lake with low phosphorus, doesn't have GBA mats. Do we know why it hasn't made it to other lakes yet? And given that Jan hasn't popped back in yet, I'm not sure if we'll be able to answer that at the moment.
I don't think we know enough about it anyway, to make that kind of answer a concrete one. So
Julian.
Yep,
Yeah, that
I
illustrates
agree. I mean, we could speculate.
the art of trying to understand these species. There's you notice them, and then you look for patterns, and then you do a lot of research, and then you try to find patterns in the research, and then eventually down the road, maybe you feel like you understand them.
And things are changing. We're seeing different patterns, with probably associated with climate change, and some other things that are happening on a large scale. So we're trying to get the big picture and learn these new patterns, but it's a work in progress.
Absolutely.
Would you mind, Erica, I'm not sure if the person is still on or not, but I have a I'd like to share my screen. I have a photos of some non-algae that looks like algae from another
Sure.
tunnel.
Yeah, go for it.
In
And it
case.
looks like we got Jan back too. So,
Oh, okay.
but go ahead, Julian.
Close this out. So the person that was asking about the algal globs, somebody mentioned bryozoan clumps. This one in the center here is Pectitinella. You can find that in several lakes, although you often have to look for it. This is Ophidium down here. It's often on the bottom of lakes and it tends to bloom in the spring. It's not algal, but it does have algal symbionts in it. And when those start to get older and mature, they'll often rise to the surface and they'll get pushed with the current into bays. So you could see those as well. So I just wanted to show a few of those. In case that answers. These are freshwater sponges over here on this side, which a lot of people don't realize we have, but they're there. Thanks.
Algae photos are so cool. Thanks for sharing that, Julianne.
Yeah. Oh, I do have one picture of what might be those clumps of, maybe I don't. If you can see here, often these kind of clouds of green, that's what happens when this benthic green algae moves off of the bottom and starts to float in the lake. So that's often what you see is large algal clumps. And I'll leave you there.
Thank you. I'm not seeing any additional questions in the Q&A box. It is 7:30, so make this your final call for last minute questions. There are some additional resources dropped in the chat. There's an upcoming Inland Lakes Convention happening in Grand Rapids this fall. That could be a really great resource for folks who want to continue conversations like these. And Erica has the link in there. And if you are interested in Introduction to the Lakes, the program that's offered by Michigan State Extension, there is a fee associated with it, but many scholarships available. So if you're interested and cost is a challenge, definitely reach out. They are probably going to be able to hook you up with a way to participate. And looks like there's a question about starry stonewort that's being addressed in the chat a little bit.
And then I just wanted to share our poll results. So, so thanks for everyone that participated in the poll. It is false. Algae are generally not a problem in freshwater environments. So as you've heard, they provide a lot of oxygen. They provide a lot of food for many other organisms. They're the base of the food chain. So they do have many, they're a critical and important part of our aquatic environments. It's just some of the ones that we're highlighting today are ones that can be problematic when there's large amounts of nutrients, and they can have some of those impacts that you've heard about today. So we hope this was helpful for you in learning more about algae, and we do want to bring you more programs like this about algae and about harmful algal bloom. So if you have questions, we'll be sending out a survey as well as the recording after this presentation. So please go ahead and share your thoughts with us about what else you would like to learn about in a future webinar, and share it with people that weren't able to attend tonight as well. We'll be sharing those YouTube links. And everyone is right, nutrient levels do affect how algae grows in a water body. So seem to have got that one right on the head. So excellent work. You can also check out in our HABS 101 webinar, the last talk was about how to reduce nutrient levels to prevent harmful algal blooms, and can also reduce kind of nuisance algae that we're talking more about today. So feel free to check those out as well. Feel free to reach out to any of us with MSU Extension, Michigan Sea Grant. If you have further questions, just going to double check and see if there's anything else that we needed to address. I don't believe so. So thanks a lot, everyone, for your time and for your participation. We hope this was helpful, and we look forward to your feedback.